Good morning and welcome everybody. We're going to wait a couple of minutes just to let remaining attendants to join us. So please keep online and we will start as soon as possible in a few minutes, 2 minutes, no more. Because are still people join us, as I said. So I think that there are still people joining, but I think that we are going to start. So good morning, everyone, and welcome to our Condalow Talk Innovation Series. We are so happy today because we have with us once again. Today we will have to talk something different from what we have used to. We are going to talk about our new product line. So first of all, I'm going to introduce myself. I'm Jaime Martinez, product manager at Condalow, and today is with us also our colleague Jose Donoso. Jose is the scientific head of our R&D department in our cell biology department. In its commitment to innovation, we create this new department less than a year ago to develop and generate value-add products for cell cultures. Welcome, Jose. Can you tell us a little about yourself and your experience?
Hi, Jaime. Thank you for your the introduction. So, so I will explain briefly my experience. Basically, I've been working always focusing in the bioprocess field. I developed most of my career in the UK, in the Oxford area. So I have 7 years experience working in a different pharmaceuticals and biotechnology companies. Yes, mainly focusing the production of the recombinant proteins as a target for research of for therapy like recombinant antibodies. And now I started this new adventure here in Spain, so I'm quite happy to come back to Spain.
We're also happy for that.
Yeah, thank you. Thank you very much for having me today.
Okay, thank you, Jose. Today talks have two different parts. The first one where I will explain the advantage of using cell culture, the importance of endotoxins-free culture media, and how Condalow can help you to achieve better results. And to support this later, Jose will present all the studies carried out on our Condalow peptones and the obtained results. So, well, once introduction were done, we will turn off the cameras and begin with the speech, okay? So, thank you, Jose. We will see you later. And also, can you confirm if the screen is seen?
Yeah, the screen is good. Yeah, I can see it.
Perfect. So, as I said at the beginning of my talk, I want to talk about the cell culture. Then we will talk about endotoxins and how Condalow can support us on this technique and this kind of culture. Well, so the first question is why cell culture? Okay, so traditionally drugs have been small molecules called SMDs, small molecule drug. Its large-scale production was done through chemical synthesis and massive screening for the search of new drugs. But over the time, limitations appear looking for these new drugs with traditional methods. Also, the complex disease, which cannot be treated with traditional drugs, were another restriction. Therefore, the need came up to find new alternative therapies with activity against this complex disease. The first option, obviously, was to use the previous experience producing proteins in bacteria. Since molecular biology techniques allow the production of eukaryotic compounds in prokaryotic cells. But once again, there are limitations in the use of bacteria to produce eukaryotic compounds. Why? Because bacteria don't always secrete, for example, the compounds of interest to the media, and also the post-translational modification is essential in the activity of the molecules produced. These proteins present different glycosylation in their structure, which means that not all molecules will be active. Human and mammalian cells usually die shortly after being removed from the body. For this reason, oncogenes were introduced into this cell that will make them immortal. Also, they can be modified like overproducer or to produce the protein only when it is intended. There are several widely used immortalized cell lines such as CHO, ELI, EC293S, and more. All of them, maybe the one with the widest distribution and use within the biopharmaceutical industry, with almost the 70% of the culture of the cell culture uses are CHO cells, CHO. And in this kind of cells, the application note that Jose will present later in a moment were also made. Of course, the most interesting part of Condalow will be this, of course, these studies and the data that Jose and his team obtained in the lab. So once we understand why we can use the cell culture, where do we grow our cells? Well, as we do with bacteria, we need a culture media where growing these cells. Also, in this case, growing mammalian cells is because they are so sensitive to change and environmental variation, and not to mention the need to control the waste product that its metabolism generates and its interaction with the culture media. The formulation of the media is also much more complex, with basic elements such as water, glucose, amino acids, vitamins, growth factors, cytokines, etc. This that we have described will constitute the basal media, the basal medium. From there, we can differentiate two large groups of culture media. We speak about complex media and chemical defined media. Complex media presents a natural source of biomolecules provided usually by fetal bovine serum, so their characterization will not be complete. I mean, one of its components are unknown. They have a wide application and they are used in the industry because they have a lower cost compared to other ones, but also getting always a very, very good performance in this case. Of course, we have said that the cell culture are very sensitive to variability. So using fetal bovine serum, okay, we will get a really good performance and it's very, very useful. But using the fetal bovine serum, we are providing a lot of variability, as we will see later. On the other hand, we have the once again, here. On the other hand, there are chemical defined medias. These media have a synthetic source of biomolecules we are added in known concentration, providing a complete characterization of each one of the components present in the media. Therefore, we will control the procedure, removing variability sources and getting optimal results. It's true that in this case, due to the high cost of this kind of media, the use is very restricted, I can say. Even so, the benefits using CDM are many, are a lot. And more if, as we will see later, if we add Condalow to our culture media. So, as we have seen, complex media are the most widespread used in the industry due to the many advantages it offers: high nutritional properties, cell protections, presence of hormones and growth factor, and many more. To get this, we need to use fetal bovine serum as a supplement to the basal media. This FBS provides all the necessary elements for the development of our culture, but has some disadvantages, such as the great variability between batches, for example. But even more important is the possibility of introducing elements that put cell viability at risk, such as viruses, prion, or endotoxins. The last ones are so difficult to control because they have a great stability, are very toxic, and can even appear in final products, putting also the health of the patient at risk. Therefore, its only presence also makes the downstream more difficult. Sterilization, deperoxygenation, filtration, etc. will jeopardize the integrity and effectiveness of the final product. To avoid the use of fetal bovine serum and all its inconvenience, serum-free media has been developed. These are culture media where FBS is replaced by other components of known origin, which introduce less variability and ensure the safety of the product, of the procedure. Among them, for example, we can add protein nitrosylate, what fit amino acids, peptides, and other components to our culture. In this way, we will be able to ensure lower concentration of unknown components and possible source of contamination. Although it is true that the maintenance of the culture media, or the cell culture, becomes more complex with this serum-free media. So we talk about endotoxins and what are they and how do they affect my culture. This is one of the main threats in the cell culture management, as is the presence of endotoxins. Endotoxins are fraction of the lipopolysaccharide present in the outer membrane of the cell wall in gram-negative bacteria. These bacteria are present in large numbers in the environment, on surface, in raw materials, and so on. Once these fractions are solubilized, they become toxic to the body, producing a strong immune response. These molecules have a small size and a great stability. They even endure some deperoxygenation processes where reach up to 400 degrees in some cases. Therefore, these characteristics give it a bioaccumulation effect through the whole production chain, and making them enemies of our bioprocess or culture media. Removing or controlling their presence can be the successful guarantee to get an optimal and safe process. And what effect do these toxins have in our culture media? Well, its presence causes that our cells develop abnormally, has a negative impact in the cell growth and product expression. It's not only cells that are at risk from the presence of endotoxin. If we're unable to remove them from our final product, they can be transmitted to the patient or final user, putting their health at risk also. So these endotoxins are elements to be considered, definitely. But how do we control them? Well, endotoxins are tested by LAL method. This method is based on the lysis of amoebocytes in the presence of endotoxin. The biological meaning of this technique is born in some very special animals, the horses crabs. These crabs have an open circulatory system that baths their entire body in blood and lymph. So everything is communicated in the body of these crabs, and minimal infection will be horrible for their life. To fight against that, the cells, amoebocytes of this peculiar animal have developed an also peculiar strategy. They are very, very sensitive to the presence of endotoxins in such a way that when they met this particle, they lyse and coagulate, isolating the treating side of that clot. Well, this technique is one that we apply at Condalow for the detection of endotoxin in our peptone, and which is included in the main international standard like USP, the American Pharmacopoeia, and IP, the European Pharmacopoeia. But in addition, we do not use the classic Hell method, which is an almost qualitative technique which allows sensitive. We're using LAL with a turbidimetric technique. Thanks to the presence of an enzymatic reagent, amoebocytes lysis produce turbidity into the sample and support it in stretch standard. We can determine the number of endotoxin with very, very good sensitivity, sorry. But more of that, we also incorporate a beta-glucan blocker. Beta-glucan is analogous molecule that are very, very similar to endotoxin. So adding this blocker, we will avoid possible false positive. So, well, finally, we will be looking for where we present Condalow. What's better than this? Great to see this great video to explain what Condalow is and how it's working. I don't know if the video will be heard, but there are subtitles, so I guess you can also follow it. Anyway, you can see the video in our website or following us in our social networks. Okay, so I'm going to put on to play the video. Meet the revolutionary toxin peptones by Condalow. These are fractions of Okay, teaching, right? Oh, okay. So are the same points that we will see before, but maybe better explain it. Anyway, what point will we highlight about Condalow? First, and as we have seen, its importance, a very low concentration of endotoxin. less than 200 endotoxin units per grain, which places Condalow with one of the lowest values on the market. Using Condalow peptones, you will be sure that your process or cell culture will be safe, and you will get the highest possible performance. We continue talking about an exclusive and revolutionary production process. As you already know, peptones are enzymatic digest of protein matter of different origin. After digestion, they are filtered to eliminate different residues can appear in this step, but maintaining many biomolecules which enhance the nutritional capacity of the peptide. And how and why is an ultrafiltered peptone different? Because to eliminate endotoxins, the filters used are much more restrictive in terms of diameter of the particle. And as we said, these endotoxins have a very small size. This means that through ultrafiltration, many of these endotoxins score will be removed, will be eliminated, but together with other important compounds for cell nutrition. Thanks to our process, our developed process by Condalow, in which we avoid ultrafiltration, sorry, but where I cannot give you much detail, we have arrived at Condalow. Peptones with the minimum concentration of endotoxins, but keeping all nutritional properties. Moreover, this process allows us to obtain great consistency between batches, obtaining higher reproducible values between production batches and ensuring to the client the least variability in the use of our products. We currently have Condalow of soy, casein, and meat from beef, all in a single 25 kg packaging. Obviously, we have sample packs, sorry. So if you want to run a pilot test and see how Condalow will change your cell culture for the better, please ask for it. So, do you think it's interesting? Do you think it? Do you find it? For sure, yes. But all this would not be possible with our R&D department and all the studies they have carried out to obtain these results. I leave you now with Jose Donoso, who will explain some of these studies cases and all data that we have obtained. Jose, wherever you want.
Okay, thank you.
I'm going to let you the control.
Okay.
Give you the control. Yes. Can you?
Yeah, can you see my screen?
Yes, yes, of course.
Okay. Okay. So thank you, Jaime, for the presentation. So we'll let you introduce myself again, just in case somebody join us during the presentation. So I'm Jose Donoso. I'm the R&D manager of the cell biology department here at Condalow. So we just basically set up a new cell culture lab, and I'm very happy to present to you the data we generated during the last months. In this case, we are going to focus in the application notes that we did, and all of them are published in the Condalow web. So Condalow web, sorry. So we're going to focus mainly in the Condalow peptones, and we're going to talking about 4 application notes. They are available, as I mentioned before, and the website. So basically, we're going to talk about Condalow soy peptone, Condalow casein peptone, and Condalow meat peptone. So this is the first application note that we generated. This is talking about the Condalow soy and the properties of growth and viability. We mainly focus in the CHOS cells, and then I will explain a little bit why we focus in this cell line. Just I would like to highlight that this is these materials and methods are going to be constant during the presentation. Basically, we use the same material and methods for the generation of the 4 application nodes. Hopefully, with our experience, this is useful for you at the time that you ordered your Condalow, and when you get this Condalow into your lab, you have an idea, or you will feel more confident handling this kind of sample. So as the presentation, this peptones comes as a powder. So it's very useful because you can dissolve this peptone, these Condalows, in your cell media of interest. So I will start, for example, in our R&D department, we work with a stock solution as we work with a small, medium scale cell culture. So basically, we took the peptone, we dissolved it in our cell media of interest, and basically we frozen down. We test a few amounts of concentrations of product. So basically, we test 20 to 100 grams liter. It's totally fine. We frozen down and then defrosted. The performance is good. So I want to clarify here. I say sterilized, not 0.22 micrometers filtration, sterile filtration. This is only to avoid the risk of contamination during the handling of the samples, because sometimes, in the cell culture lab, it's quite difficult to keep contamination away. Nevertheless, we did some test about this, and we just test like a very high concentration of peptone, Condalow peptones, and we plated in LV agar plates, and we didn't see any bacterial growth. So probably it will be safe to use this Condalow straight to your large scale cell culture. So why CHO cells? We used CHO cells for the whole presentation, and for the whole application notes. We thought, it's the one with the more interest, as is being used worldwide for the bioproduction or the production of different biomolecules as a therapy or as a target for research. So we are hoping to get some more new cell lines and produce more literature about these peptones. As advice, probably when you receive the samples, they are not ready to go. So possibly you just need to test a few concentrations for your cell culture. Because obviously, as you know already, different cell lines, they will have different metabolism, and they will use different amounts of different nutrients. So probably you will, what we did is basically test a panel with different concentrations from 1 gram to 10 grams liter. Focusing more in our presentation, basically we use basal media, serum-free media, chemical defined media, that long name, as a control. We did we took samples for this cell culture every day during 5 days, because we noticed that from the 5th day onwards that the cells started to lose the viability. And bear in mind that we always, as we're talking about numbers of cells here, we're talking about viable cell density. So this is something important to have in mind. So also to mention that we used for this counting these cells, we use an automated cell counter, which is worldwide use and is very famous, and everybody probably have one automated cell counter like this. So it's very reproducible with the rest of the data in the world. So let's start with the first graph. We are going to focus on the effect of Condalow soy peptone on CHO as cells growth. In this case, we can see here on the left the viable cell density from 1 million to 9 millions, and then here we can see the days the samples were taken to measure the viability of the cells and the numbers of cells. You can see here a little bit of Spanish. Well, I think nowadays everybody knows a little bit of Spanish. So dia is day, okay? So and focusing in the graph, we can see the control here. Basically, if we remember, our control is the chemical defined media for CHO, for this line, for this cell line. And here you can see the Condalow peptone, which is the Condalow soy peptone. Basically, we add 1 gram liter of our Condalow soy peptone to this cell culture media, to this chemical defined media. And you can see here is like a striking difference between the growth of the control and the control plus the 1 gram liter of our Condalow soy peptone. So basically you have with the control a performance at the 5th day of 4.5 million cells by viable cells, and then you have here just adding 1 gram liter of our Condalow soy peptone double. Basically we managed to double the number of the cells. This is quite good because if you have a very expensive chemical defined media, just by adding 1 gram liter of this Condalow soy peptone, you increase the performance of your culture a lot. The yield is much more higher. So moving into the next graph, we can see here on the right the batch-to-batch comparison. As scientists, we everything that we do in the lab, we would like to be reproducible and robust. And sometimes we know that in cell culture could be a little bit tricky, and especially with this kind of material like hydrolysates and peptones, as they are from natural sources, it could be a little bit variable. So that's why we wanted to do this test in the lab. Basically, we follow the same scheme. We have on the left the viability of the cells, number the cells milliliter, and then we took the samples every day during 5 days. And this was counted in the automated cell counter. Here again, the control is just the chemical defined media for these CHO cells, and then we add here 1 gram liter of Condalow soy peptone of different batches. Basically, these batches were produced in a different time, following the same procedure of production. As you can see here, the variation between the assays at the 5th day is not much. It's very robust, and you can see that you can find out the same kind of performance during different batches, which is quite good and quite special about these peptones. Now here in the next application note, we wanted to focus how is the performance of our Condalow soy peptone against ultrafiltered soy peptone. So basically we wanted to compare the performance of our way of producing this Condalow against the wide use ultrafiltration method. We use the same material methods as mentioned at the beginning of the presentation. And now if we focus in the graph, basically we follow the same scheme. It's nothing new. So we have the viability of the cells milliliter on the left, and then the days on the right, 5 days. Basically we have 2 million to 10 million population here. So as you can see here, the control again is our chemical defined media for this CHO line, cell line, and then we have the ultrafiltered peptones here. And then on top of that, we have our Condalow soy peptone. So as you can see here, I think with this application note, we can see nicely how our Condalow peptone, Condalow soy peptone is performing very similarly, even better than the rest of the ultrafiltered peptones. So basically, with the way of fabrication of producing these peptones, we managed to keep all the macronutrients that otherwise will be eliminated with the ultrafiltration. We keep exceptionally very low endotoxins levels. So as a wrap up for this Condalow soy peptone, I think it's quite interesting to highlight that only with 1 gram liter, you can see a really good performance. We almost doubled the cell density, viability as well. I mean, the viability of these cells is, I mean, they're viable cells basically, sorry. And then basically we double the number, only adding 1 gram liter, which is quite good. And also this Condalow soy peptone is animal-free and GMO-free, so it will be good to use in to produce different therapeutics if you are interested in that way. And also I think is another thing to remarkable is their reproducibility, and it's very robust. So I would say it's quite good product to have in the lab to increase your yield for the cell culture. So the next application node, it will be casein. So we wanted to focus again how this casein peptone performs against the different casein peptones. This is something interesting because we couldn't find any ultrafilter casein peptone. So as again, we're going to go straight to the graph because the materials and methods is the same as we used before, and everything will be very described in detail in the application notes in the website if you are interested. So we follow the same scheme. We see here the number of cells, the viability of the cells, viable cells again, and then the days that we took the samples. Okay, and then here we find out that the control, which is the chemical defined media, start to plateau at from day 5 to day 6, and our Condalow casein peptone is performing very well. It says around 20 millions, which is quite good performance. And here, as I mentioned before, we couldn't find any ultrafilter peptones, casein peptones. So I imagine what is happening here is this no ultrafilter peptones. They have some certain levels of endotoxins. So this is quite interesting to see because you can see the effect of endotoxin directly into the cell culture. It's performing like in a worse way than the control, and it's very far from the Condalow's casein peptone. So I think this is, for example, as a sum up for this product, I think this casein peptone, this Condalow casein peptone, has an exceptional performance. We did several trials, and we see that this is one of the best performance that we have. I think it's unique because probably you cannot ultrafilter casein peptones. That's why we suspect, because otherwise we could find some of these products in the market and we couldn't find them. So I think it's a very special product to increase the yields of your cell culture. This is the last application node that we generate. This is talking more about Condalow meat peptone again versus different peptones in the market. So materials and methods are the same, and we follow the same scheme for the graph. Basically, the cell viability density, the viability cell density against the days which the samples were taken. Again, the control is the chemical defined media, and then we basically use the same concentration for all of them. Even in the casein, I forgot to mention, but we use 1 gram liter of the peptone. Here again, we see the comparison with the different products, and we can see that the non-ultrafilter peptones, they have a very good performance, surprisingly, but I will not trust this data because probably if you, for example, you buy this no ultrafilter peptones, you will end up having some issues for sure, because the batch to batch variability would be very, very high. And then also we here again we see as the Condalow meat peptone on top of the performance of all the peptones, even the ultrafilter peptones. So I think as a sum up, I would say this Condalow in general, I mean, this Condalow meat peptone is quite good. I mean, it keeps the macronutrients and they don't have any of the endotoxins. So that's why always is one of the best performance in our assays. So I would say, in my experience working with these Condalow peptones, my conclusions are that this Condalow allows your cell culture to reach higher BCD, viable cell densities. I say that correctly this time, than the chemical defined medium on its own. Only with 1 gram liter, you can have a really good performance, almost double it. So we saw like a better performance always. So we never saw any Condalow going lower than the control. Condalow's elaboration process, I think I mentioned before, they maintain all the benefits, the beneficial nutrients, and the hydrolysate keeps the remarkable low endotoxins. So we saw in this few application notes that this product behaved very, very good because we had an hypothesis that we thought that we could keep the macronutrients and have a better performance and also keep the endotoxin levels, and it's working as sold. Condalow offers a wide range of solutions for your cell culture, like animal-free peptones. I guess the Condalow soy peptone is probably one of the more interesting ones, but also we cannot forget about the casein and meat peptone. They have, especially casein, they have Condalow casein has a very good performance in BCD. So I think it will be a good thing to at least test in your lab. And obviously, I mean, as mentioned before, the cell culture lab is very recent. So we are working to expand the knowledge of these peptones. We are gonna prepare some more application notes with the different characterization of these products. And in the future, we would like to expand our cell culture portfolio. So that's me. Thank you very much.
Thank you to you, Jose. Thank you for all your work and knowledge about cell culture and all your support in order to develop this new line. Thank you so much. We have some minutes to answer some questions. I'm happy because we have many questions. So I also at the same time I'm a little bit sad because we don't have much time. Anyway, don't worry because we will respond and answer all the questions that you made by email. Anyway, also if you have any kind of question, you can write us to our mail. I'm going to share the Jose, can you give me the control, please?
Yep, no worries.
As I said, you can write to micro@condalab.com is our main mail. Okay, thank you so much.
No worries.
Now, here, right? So, well, we will wait with the first question. Someone is asking about in what kind of media they can add our hydroxylates, our Condalow peptone. I think that's obviously the CD and serum-free media, but maybe I don't know if you have other information, knowledge about using Condalow maybe in complex media.
Yeah, well, we at the moment we had time to do to test with chemically defined media, and that it works very well. And also we try with some basal media plus serum and also the peptone that didn't work very well. But I think this is very complex because probably for the best it will be to get some samples and test for yourself in the lab, because as we know, different cells, they have different metabolism and it could be very different from one case to another. So I will recommend
depending on the application, of course, also kind of cell they are using. Maybe the better in this case always is to test the samples, because maybe they will get very good results.
Yeah.
On this line also we have other question that I think that is very interesting, because they are she's asking about, are biopharma manufacturers using chemical defined media or complex media nowadays? What is the trend, the adoption rate, and the percentage of the biotherapeutics made from either types of media? I think that maybe the more uses right now could be the complex one with MBAs, but we really realized that the trends on the biopharmaceutical industry, and Jose was working on that, is to use more and more the chemical defined media, right, Jose?
Yeah, I think the trend is clear. So they're moving to animal-free chemical defined media. So probably a good way to start to move to that world will be some probably ordering the chemical defined media with some hydrolysates like Condalow soy peptone. It will be very interesting. But I think probably, yeah, they're moving into that way. So they want to eliminate any risk, and they want to control all the processes.
The variability, and I think maybe on this kind of processes and manufacture, a little bit difference in the final performance, the final yields, will increase a lot of in the money with the final product. So for the pharmaceutical industry, they are very interested to have everything controlled, everything with the less variability possible, and then there is these trends to move to chemical defined media. But also the new biotherapeutics products, of course, the biotherapies for cancer, for immunosystem, autoimmune, disease, all this kind of new approach to new drugs are made, and they are studying with cell culture, I guess. Okay, so there is other, it's also, I think that you talk about it, Jose, but people is asking about if we are going to test, not to do more tests with Condalow, of course, but if we are test Condalow in other cell lines.
Yeah, the one of the objectives of the department is to basically test the most cells that we can with the new product, with this Condalow peptones. We are working in the 293F HEC cells, also Bero cells. So we're trying to increase the portfolio here, and then probably characterize even more these Condalow peptones.
Also, people know that it's a baby department because we don't have more than a year, so we have a very long path and way to work. So perfect, and for also it's time to be close. But anyway, we're going to answer the last question. Is about the packaging, because people is asking about there is only the 25 kg pack. At the moment, well, of course, the product is available in 25 kg. Also, there is 250 grams for a sample format, sample packs. But anyway, as you already know, Condalab is a very flexible company. We are always supporting to our customer. So anyway, if you have other kind of necessity to pack in order maybe 10 kilos, 1 kilo, wherever, just to say to us, and we will try to do the best from outside. So there is other questions that will be answered by an email. No worry, all of them will be answered. So I also thanks again to Jose to stay with us today. Thanks, Jose. And also thank you for everybody that is joining us in this Condalab Talks. I hope to see you soon and have a nice day. Bye-bye.
Bye.
Bye.